students, in the last two videos, we are studying about the features of ideal vector. So there are four features of ideal vector. The three we have discussed. So in this session, we are going to study fourth feature of ideal vector, that is the vector it is inserting into plants and animals. Inserting into fourth one, our DNA is inserting into plants and animals. Here you can understand one of the bacteria or a pathogen, it's Agrobacterium tumefaciens. It causes cancer in dicot plants. It has one of the specific DNA. It is called tDNA. tDNA. So by inserting this tDNA, it causes cancer in dicot plants. The another one of the virus, it is called retrovirus. Retrovirus, it causes cancer or it transfer normal cell into tumor cell in animal cell. So Agrobacterium tumefaciens, it causes tumor in plants, in dicot plants, but retrovirus, it causes tumor in animal cells. So both are pathogenic. It's harmful. But now here, by the help of this biotechnology, these both are disarmed. Agrobacterium tumefaciens, it is disarmed. tDNA, it is disarmed or it is modified into TI plasmid. Modified into TI plasmid. It means tumor inducing plasmid. It is modified as our gene of interest and it is inserted into the plants. So it is modified and it is inserted into our gene of interest and it is inserted into the plants. So because of modification, genetically modified, so now this agrobacterium is no more pathogenic in plants. Same like that in the retrovirus. So in retrovirus, it causes cancer in animals. So that also disarmed. It's not pathogenic. It's modified that such gene into our gene of interest and it is inserted into animals. So it is no more pathogenic. It's not more harmful to the animals. So this you can understand. This is also one of the feature of ideal vector. So now our plasmid or the vector is ready. Now our product is ready. What's the next step? We have to sell to the customers. So if that we have to make the plasmid to competent, means it should be attracted by the customers. So here the customer in biotechnology, the customer is host. So we have to make competent host. See, actually what you can understand here, cell is hydrophobic. Cell is hydrophobic, but DNA is hydrophilic. Here the question is how to make competent host. This is one of the cbse board question as well as the NEET question, how to make competent host. Before studying the answer, you can understand cell is hydrophobic and DNA is cell is hydrophobic and DNA is hydrophilic. So what? So both are against, it is negative. But we have to insert the DNA into the cell, into the host. So the first process you can understand, we have to treat the cell with divalent cation. Treat the cell with a divalent cation such as calcium. So what happens? Concentration of the pore, it will be increased so that we can insert the DNA into the host. So increasing the concentration, treating the cell with the divalent cation. That is one method. The another method you can understand, heat shock method. It is called heat shock method. So what happens in the heat shock method? First the cell is treated with, it is cultured with more temperature, that is 42 degrees Celsius. It is increasing the temperature. After that again it is decreasing the temperature. Again increasing the temperature. So by the help of heat, increasing temperature, decreasing the temperature, that only you can understand the word, it is heat shock method. Because of that the concentration of pores will be increased. Immediately we can insert our DNA into the host. So that you can understand the second method. And third method you can understand, it is electroporation method. Electroporation. So by the help of the electricity, by the help of the word itself, you can understand applying electricity, increasing the concentration of pores so that we can insert our DNA into the host. So in this way we can make competent host. Now we are going to study, there are three methods, that is called vectorless method. There are three methods, it is called vectorless method. What we have studied means actually in the biotechnology we need vector for inserting our gene of interest to the host. But here you are going to study vectorless method. So first vectorless method is microinjection. And the second one is called gene gun, and the third one is called disarmed pathogen vectors. So these are the three vectors of three vectorless method. So we can study one. The first one is microinjection. In the microinjection, directly the DNA is inserted into the animal cell. Directly the DNA is inserted into the animal cell, that we can say microinjection. It is vectorless, directly inserted. Now the gene gun. It is also called biolistics. It is used for the plant cell. Directly it is bombarded with high velocity particles like tungsten and gold. It is bombarded, DNA is bombarded, and directly it is shooted to the plant cell. That we can understand gene gun. So here it is animal cell and this is used for the plant cell. Now the third one, disarmed pathogen vectors. It means directly the cell is infected, particular our gene of interest, that cell is infected to the plants or animal cells, so that we can transfer the cell into the, we can transfer the DNA into the host. That we can say disarmed pathogen. So these three methods are called, this is also one of the important question. It is called vectorless method. Thank you students.